So
Which one?
just,
This one?
that's the pointer.
Very
There
good.
you go. And then you
And
can
then
advance.
Advance this. Okay,
Advance forward
perfect.
and back.
Yes, yes, yes,
There we
that's
go.
great. I
And
have.
let's get you organized here.
Yeah, I think that's fine.
And we're going to tilt you down just a little bit there.
All right.
And we're going to be fine.
Thank you so much. It's still good morning for me. I don't know how you feel. And I thank Darius for giving an excellent talk. And it will be hard to follow him up, but I'll do my best. And what I want to cover during my talk is what's cooking in the kitchen and what's cooking in the lab. So I would like to tell you the new research that we're doing, the new discoveries that is coming up, and how we think that's going to change the translational research and also expedite the transformative or the translational personal therapeutics to upper motor neuron diseases. And I put the title as Building Effective and Long-Term Treatments to Upper Motor Neuron Diseases, and I really think that time is now. And it's not that we are waiting for things to happen. I think things are happening, and I think this is a very exciting time for upper motor neuron disease research. So we always ask to ourselves, like, why there is still no treatment for upper motor neuron diseases, right? Why there is no treatment? And I put them in three lines, and one is that we did not know why upper motor neurons become vulnerable and why they degenerate while other neurons in the brain stay relatively healthy. Okay, so the why is the reason for selective vulnerability. And then how can you understand this, right? Are there good model systems that recapitulate human condition? The data that we generate, can it be translational? You know, that was a big missing piece in the equation. And then the second is there's really no effective drug discovery for upper motor neuron diseases. There are no drug discovery platforms using upper motor neuron health as a readout, so we don't really have biomarkers, outcome measures. Even if we had a drug, we would not know if it worked in the patient in a clinical trial setting or not. So there were many unknowns and there were many lacking pieces for building a drug discovery for upper motor neurons. Diseases, and also remember that it's a rare disease, right? So there are clinical challenges, as Darius has mentioned, but there are also technical and biological challenges. And then because the disease is rare, it's heterogeneous, but it's pretty much cell type specific. It's really the upper motor neurons. So can we really develop a gene therapy? Can it really be that we modulate the genes that are mutated in distinct patients? But how do we deliver the genes to the right neurons? Because brain is so heterogeneous, but only a select neuron population becomes diseased, you don't want to give the gene to every neuron, every cell, because while trying to fix something, you may actually initiate many other problems. So the selectivity is also very important, and we didn't know how to do that. So, you know, these were mainly the three major problems, and now I'm going to go over them individually and try to show you what progress we have done over them, okay? So why upper motor neurons degenerate? So this is a very, very fundamental question. Upper motor neurons are located in the motor cortex in layer 5, and they are a very unique neuron population. They project to the spinal cord first, right? They have an input to the spinal motor neurons, and the spinal motor neurons project to the muscles and, you know, the tendon, like to the periphery. And all voluntary movement that requires us to think, like, you know, cognitive movement or voluntary movement, requires input from the brain. And if you look at these neurons, they have a very large cell body. They have a very long apical dendrites. I'm going to come to that. They have, you know, this is the basal dendrites. This is the apical dendrite, and they have a very, very long axon. Imagine that the cell body is here about, you know, 30, 40 micrometer, but the axon could be a meter long. So the length of the axon is very big. But these neurons degenerate in the brain. Okay, and we are trying to understand why they degenerate. So we have to have good model systems. And one of the model systems to understand neurodegenerative diseases is the transgenic mouse models, humanized mouse models. So mouse models are pretty much used very effectively. But the thing is, you can't compare apples and oranges, right? So humans are not mice. So we can't really learn directly from mice. Like, you can extend the survival of mouse, but we learned that that does not translate to clinical trials. Like, they were expanding the lifespan of SOD1 mouse, but when you go to clinic with ALS patients, that didn't work. So I think comparing mice to human at a species level may not be feasible. But if you look at the upper motor neuron in the mouse brain, and if you look at the upper motor neuron in a patient, at a cellular level, they are very much identical. Like if I gave you 2 slides and I took images of the upper motor neurons taken from a mouse brain and an upper motor neuron taken from a human brain, despite the size, because human is of course much larger, right? You may not be able to tell which one is which because they are still pyramidal, they have large apical dendrites, their location is the same and so forth, they have spines, they have branches in the upper, so it's very much similar. But then if you look at electron microscopy inside those cells, okay, so that you begin to see their mitochondria, you begin to see their endoplasmic reticulum, you begin to see their nucleus, there's no way you can tell the difference. Impossible. They're identical. The mitochondria, let's say in an upper motor neuron in a mouse, is exactly the same with the mitochondria that human patients have in their upper motor neurons. Same with endoplasmic reticulum, Golgi, nuclear membrane. So all the inside organelles, the structure of the cell is identical. So then when we want to build translational efforts, we need to go down the denominator to find common denominator, right? And we think at a cellular level, the upper motor neurons, to understand the upper motor neuron, we really focus our attention to the cells because the translation, we think, is at a cellular level. The other thing I want to emphasize is patients come to clinic because they have a phenotype or they have a clinical manifestation. But before that clinical manifestation occurs, the motor neuron circuitry begins to malfunction or begins to dysfunction, or there's something wrong with the motor neuron circuitry. And if you look at the motor neuron circuitry, the movement starts in the brain, in the upper motor neurons, right? So the neurons become diseased or become unhappy even before motor neurons circuitry becomes dysfunctional. So the order is cells become unhappy, motor neuron circuitry becomes, I don't think I can do this, it starts having some problems. Then starts having so many problems that patient starts showing symptoms and then the patient goes to the doctor. So the initial or the original problem actually arises much, much earlier than the patient goes to the clinic. And it is inside these cells and inside these neurons. And we find that there are common problems. And I think to have effective treatment strategies, we really need to focus our attention to the culprit, like to the one who is responsible for the problem. Okay, so now I'm going to talk a little bit more about mitochondria, ER, and let me give you a summary. And mitochondria is an organelle that we have in our cells, especially in upper motor neurons, and they have a very unique structure. This is the outer membrane, and here is the inner membrane. And the inner membrane of the mitochondria needs to be intact because for it to produce ATP, the energy, the inner mitochondrial membrane has to be intact. And the DNA of mitochondria is also in this area, which is matrix. And if this region is broken, then the DNA leaks out to the cytoplasm, and that's also one of the reasons that neuroimmune modulation is initiated. And mitochondria is also important for the storage of calcium and other ions, the communication with ER. But mainly, it's important for energy production, metabolism, and so forth. And look at this. If you look at the upper motor neurons in human, this is an ALS patient which show prominent upper motor neuron loss. Here is a normal mitochondria. You can see the cristae, the inner membrane. And in the ALS patients, there's almost no inner membrane. It's empty. So that's a big problem. That means they can't generate ATP. That means metabolomic problems and so forth. So then if you look at the upper motor neuron in the mouse, like say SOD1 mouse, TDP mouse, now we have spastin mouse, profilin mouse, like we looked at 5 different mouse. Again, you see all the, like even breaking here, cristae structure is gone. You can see hollow structures in the inner mitochondrial membrane. So it seems as if this structure of the mitochondria is one of the converging problems. And we published this in recently, showing that the translation is really at a cellular level. And if you look at the upper motor neurons in ALS patients with TDP pathology and also ALS mouse models that reciprocate TDP pathology, it's basically the same. So at least now we have a target, like we have to improve the integrity of the inner mitochondrial membrane. So the other major organelle in the cell is the endoplasmic reticulum. It's hard to tell, endoplasmic reticulum, you know, but it is the site where the proteins are made. And here is the nucleus, and here is the endoplasmic reticulum. It's like in sacs, right? And ribosomes are here. That's where the messenger RNA comes in. The protein is made. But if you look at normal control, you can see all these, you know, beautiful structures. Here are like the 1, 2, 3, all these sacs. And look at the ALS patients. They are swollen, they are broken. You see free ribosomes around. They can't maintain the cytoarchitectural integrity of the ER. So if your ER is like this, you are not making proper protein, you are prone to have ER stress, so there are problems, right? And the same thing happens in the ER of upper motor neurons in mouse models with upper motor neuron defects. Again, they are either swollen a bit, they are broken. You can see like itsy-bitsy pieces here and some swollen sacs here, and it's just, it's not normal. In the here, it's in the wild type, it's beautiful. So now we have two things that is problematic in the cell. And if you have problems with ER and mitochondria, and of course they also interact with each other, you have major problems with energy production, with protein production, and you know, it's very hard for the neuron to be healthy. And I did not show here in these images, but let me tell you this. If there was another neuron, which is not an upper motor neuron, but sits just next to an upper motor neuron, guess what? Their mitochondria is fine, their ER is fine. So it is very, very cell type specific. So there's something going wrong inside these upper motor neurons that makes them sick while their neighbors are still enjoying their, you know, barbecue. But there's something wrong with these upper motor neurons, they're crying. So we have to understand why they suffer. The other thing that we found that is common between the upper motor neurons in human and upper motor neurons in well-characterized mouse models is at the site of the apical dendrite. And here is the cell body for the upper motor neuron. Here is the apical dendrite, and this is the top of the brain, all right? And this apical dendrite is extremely important for the connectivity and function of the upper motor neuron, because this is exactly the site where other cortical neurons are going to speak to them. And how do neurons speak? They speak by making connections through these itsy-bitsy little spines. You can see even here in the healthy apical dendrite, there are thousands of spines, okay? So we speak with words, but they basically touch with their spines and they form a connection. And it's like electrical activity, right? And the upper motor neurons can connect to thousands of neurons at the same time. I mean, imagine in a room, thousands of people speak at the same time. I don't think we would understand anything, right? But these neurons, thousands of neurons speak to them, and they integrate that information. And they say, you want me to send an action potential to the C4 region of the spinal cord and then to this alpha motor neuron, right? Yeah, right. And then they just send the action potential very selectively to a very distinct target. And they can do that. And how can they do that? It's because their apical dendrite is very intact. And look at the different mouse models here. Look at the apical dendrites. It's filled with vacuoles. There's not even a spine left. I mean, if your apical dendrite is like this, how are other cortical neurons communicate to you? How will they tell you what to do? How will they tell you how to start an action potential? You won't be able to get an input, and you will be left out of the conversation. So then you will be just sitting there, and then nobody's talking to you. You don't start an action potential, all right? And then your mitochondria is sick. You're not making ATP, and you're not making protein. I mean, how can you be happy, right? How can you be happy? And also, if you look at the human in the sporadic ALS, familial ALS, again, look at the apical dendrites. They're falling apart. So we have to improve their cytoarchitectural integrity. We have to make sure that they have enough spines. We have to make sure that they're in the circuitry. We have to make sure their mitochondria is happy, their ER is happy, they're making proteins, there's no protein aggregation. So they have many problems. It's not just one problem, right? So we have a big task, but at least we understand what the problem is. And that was possible because we realized that the upper motor neurons, if we focus our attention to the cells, to the neurons that degenerate, they begin to speak to us, right? And we published this in here. All right. So then, I want to emphasize one more time that the translation is at a cellular level. That means when upper motor neurons degenerate, the motor neuron circuitry becomes impaired, symptoms begin to occur, and patient is diagnosed, and then the disease progresses. But if we make that upper motor neurons happy, then the motor neuron circuitry may be restored. Then the symptoms may actually improve because now you have functional and working motor neuron circuitry. And the patient may begin to feel better because now their circuitry is moving better, right? And then disease condition may be improved. So then I think the goal is to make the upper motor neurons happy. So if we can make them happy, we all will be happy, okay? So my goal is to make the upper motor neurons happy. But how? First, we need to learn what makes them upset. Then find things that cheer them up. It's pretty easy, right? All right. But then to make them happy or to understand what they like, we have to become friends with them. Like we need to see them, we need to know them, we have to isolate them, we have to talk to them, and how can we do that? And cortex is very heterogeneous, every neuron is very different. So then we were actually the first group to make a reporter line that is very specific to the upper motor neurons. So there were many reporter lines previously generated, but they're not specific to a neuron subtype. So we made a reporter line, we call this the EGFP-UCHL1 promoter line, in which not all neurons in the brain, but only the upper motor neurons fluorescent. How cool is that? And it's the motor cortex, and the fluorescence becomes stable. Even at like 2.5 years, you can still see these neurons, okay? And they're not in the corpus callosum, which means they are not callosal projection neurons. So we published this paper. I'm not going to go into detail, but we confirmed by anatomy, by molecular marker expression, by electrophysiology, by this. I mean, we killed the horse 5 times, and then we confirmed, yes, this is indeed the upper motor neuron, okay? And so now that we generated the upper motor neuron, we can see them. And this allowed us to shift the focus from mice to neuron. So then I don't care which mouse. As long as I have their upper motor neurons fluorescent, I can actually cross them with any disease model, like SOD1 mouse. We cross them. Here we have the SOD1 mouse with the upper motor neurons GFP. So then I have the fluorescent upper motor neurons that become diseased due to SOD1 mutation, that become diseased due to TDP pathology, that become diseased due to alsin. And recently, in collaboration with Dr. Bass, who generated one of the best mouse models, the spastin mouse model for HSP, and in collaboration with him, we now generated the spastin upper motor neuron model. So that we know all these upper motor neurons are fluorescent and they degenerate due to a different underlying cause, right? They all die due to different reasons. And here, for example, SOD1, there's oxidative stress, ER stress, mitochondrial defects. In TDP, it's protein aggregation, nucleocytoplasmic transport, the transport from nucleus to cytoplasm. So they represent different underlying causes and different subpopulations of upper motor neuron disease patients. So every of these upper motor neuron is a spokesperson for a subgroup of ALS or HSP-PLS patient. They develop the disease due to different reasons, okay? And we have their upper motor neurons fluorescent. How cool is that? Then, what do we do? Okay, we have the tools, wonderful. What is the molecular and genetic basis of upper motor neuron vulnerability? So then we ask these neurons, why do you become vulnerable? Tell me. What makes you sick? You speak to me now, all right? And how do we do that? We actually use FACS, which means fluorescence-activated cell sorting. And it allows us to purify the neurons based on their fluorescence, okay? So you dissociate all the cells, but only the GFP ones will be isolated. And non-GFP are the ones that are not CSMN. So among this whole complex structure of the cortex, we pick only the ones that are upset. And then you can also isolate the ones that are not upset, right? Then we do our RNA-seq, which means we look at the genes that are expressed. We look at proteomics, which means the proteins that are present in those cells. Then we do metabolomics, looking at the metabolites that are present in those cells. And we do this in many different mouse models, right? And we do this at different time points. Because what makes you sick when you were a child is different what makes you sick when you're an adult and what makes you sick when you're a teenager. So different things make you sick at different time points, right? So during disease, it's the same. So you may be upset because, I don't know, calcium, voltage-gated calcium ion channel is not working properly, how dare, right? But then the second, this protein accumulation, blah, blah, blah. So then the reasons that make you sick along the way may be different. So then that's why we also add a time scale to this to see how things change with time and also with different causes. All right. Then for the proteomics, we do top-down and bottom-up. That's also very important because for top-down proteomics, you don't chop proteins. You basically read the whole protein. And that's very good because then you can tell if there are any splice variations, if there are any post-translational modification differences, if there are any structural changes in the protein. Not that what protein you make, but what kind, like what are the what you call like very specific differences in those proteins, okay? All right, so this is a major, major undertaking, as you can imagine. Then after we do all this, all the omic data and everything, okay, for the sake of time, I'm gonna put this all together. Now we begin to understand why they are upset. And they have problems with protein aggregation, problems with endoplasmic reticulum, problems with mitochondria. They can't maintain their cytoarchitectural integrity. They have problems with axonal transport. And so these are some of the major problems that these neurons have. But based on which mutation you have or which underlying cause, let's say this is your major problem, or someone else, this is the major problem. So the extent of impact that these problems make on individual neurons may be different, but this is basically what goes wrong with them. All right, so I'm going to come back to these just to remind you the apical dendrite, the mitochondria, and ER, because we realized that these were the problem sites for the upper motor neurons. So then after we realize that these are the major problems, okay, but we are using, again, purified upper motor neurons from the mouse, right? Then we decided to think outside of the box, and I'm going to explain a little bit. Darius said, you remember, he said maybe we now know 70, maybe more than 70 genes, gene mutations that cause HSP. And the thing is, those genes, if you look at them, may seem unrelated. One of them is in the lipid homeostasis pathway, one of them is in the axon transport pathway, one of this pathway, so they are all very different genes. But imagine this, you have one mutation or you have one gene that is mutated that leads to a disease. So I bet that the protein that is coded by that gene must be so important for the function of the upper motor neuron that when that one gene is mutated, it says, you know, I can't do it. I can't do it. Like, this is so crucial to me that when it is mutated, I'll just drop the ball. Like, yeah, I can't. So then what makes that protein so critically important? So then we said, okay, how do proteins function? They bind to each other. They are within a canonical pathway. You know, they have partners, binding partners, activity partners, blah, blah, blah. So we then looked at all the binding partners, all the genes that are mutated in HSP and PLS patients. And then one protein said, okay, I bind to A, and when I bind to A, you know, we go to a pub. But when I go to B, we go dancing. So they all have different partners, but with each partner, they do something different, right? So then we said, okay, tell me who your friends are. Okay, my friends are A, B, C, D, E, F, G, okay, so the second mutated gene, tell me who your partners are, or who your friends are, whom do you communicate with, whom do you work with? And it says, oh, my friends are blah, 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 blah. Then we got all the friends together in a room, and I said, friends, what do you do? What is the canonical pathway that you are involved in? What is the cellular event that you take part in? And tell us what you like to do most. Oh, they begin to speak to us. Okay, so then this is the paper that we published. So then we made the mutations speak to us. Because we looked at all the HSP causative genes, associated genes, linked genes, and we asked all of them individually, whom do you work with? All these binding partners. Oh, yeah, this one, I bind to these are my binding partners, these are, so this list is huge, right? So we have 400, 500 of them. So we got all these lists, all these binding partners, and imagine what, there are some famous guys. For example, you know, the YWHAZ binds to this, YWHAZ binds to that, YWHAZ is a famous guy, he binds to everyone. So, do you see what I mean? So then there are some famous guys. What are those famous guys doing? And so we try to understand their interaction. And after we do that, we then say, all right, all the proteins, where are you? What do you do? Where in the cytoplasm are you? And then what are the cellular events that you like? Tell me who you are, who is your friend? You know, tell me your friend, I'll tell you who you are type of thing, right? So then they say, all right, look at this, the p-values 10 to the minus 23, minus 14, minus 39. I mean, there's no chance that this is luck. Usually we say it's a significance when it's minus 3 or minus 4. So this is minus 23. So it says, I think there's a problem with neuroimmune modulation and lipid homeostasis, cytoarchitectural dynamics. So all the friends that they work together, they make sure that there is really a check on lipid homeostasis. And there's a check on cytoarchitectural dynamics. And there's a check on neuroimmune modulation. So we all work together just to make sure that these are all working properly. All right? So this also tells us, and we learn just from the mutations. Then we go deep into lipid metabolism, for example. Lipid metabolism, what are the canonical pathways? Ooh, there are some very important ones. And show me on the map. So here's the adipogenesis pathway, for example, and all the pink ones are the ones that are friends, that are within that map. And then we say, all right, which ones are you, the famous guys, and you are doing what again? All right, so we identify, because those famous guys could be the targets, because they speak to so many people, right? And by modulating, by activating them or something, we may actually have a broader impact, not just the mutation itself, right? So we look at the downstream signaling cascade of events, upstream signaling cascade of events, and so forth. And again, growth factors, they represent, and so forth. So to this list, we also added the lipid homeostasis. So now we know the major problems, which is pretty good. Then, again, it's the mitochondrial problems, ER problems, axon transport problems, lipid homeostasis, and but of course, with different patients, it's different. Like, we can't target all in all patients. Like, for some patients, one will be more predominant. Okay, so at least we begin to understand why they become disease, and that's a major, major undertaking. The second is no effective drug discovery for upper motor neurons. And drug platforms don't use upper motor neuron health as a readout. Can you imagine, even for an upper motor neuron disease, there are drug screening, preclinical assays, they use, like, I don't know, fibroblasts, some kind of cell, like their cells or they use are not really related to upper motor neurons. And how can you learn from a cell that is not related to upper motor neuron whether they like it, the treatment or not, right? But then they tell us, it was not available before, that's the only one we have, this is easy to grow. Yeah. So then we decided to tackle that on, okay? And now that we have this reporter line where upper motor neurons are GFP, we said, can we actually, okay, and I also told you that we crossed some two different disease models, right? We have different disease upper motor neurons that are GFP. Now, they invited us to read many reviews about this in the drug discovery world, because we really think moving forward in clinical trials for ALS, we really have to get our information from motor neurons. And we want to incorporate upper motor neuron health in drug discovery. And so we are beginning to develop novel screening platforms. You can have in vivo platforms. Let's say you have the disease, and here, look at the healthy. There are many, and disease, there aren't any. You can give them by IP, by gavage, by many different ways, and then you treat them, and you look to see if you can retain the upper motor neurons in the cortex, Or you can put them in culture, and this is very important. Because when you put them in culture, they retain their GFP expression. That's very important. You know why? Because in a dish, you have dissociated cortical cells, and you know exactly which one is an upper motor neuron. So you add the drug, and you watch them. If they like, they extend the long axon, they branch, they eyebrows, they're happy. And if they don't like it, they shrink, and then they die. So they tell you if they like it or not, okay? So this is like one of my views. Like blue is the other cells, green are the upper motor neurons growing, and red is the astrocytes. So in a plate, you can actually see which ones are the upper motor neurons. And I like this one. This is a diseased upper motor neuron. Look at this. This is an astrocyte, and it's as if it's running away. So you can also see the interaction of the neurons with other cells in culture. And this, when we built this in vivo, in vitro drug screening assay at the time, Dr. Silverman developed the compound NU9. And Dr. Silverman is the inventor of Lyrica. He's at Northwestern. Then we teamed up to see whether NU9 would have an impact on the diseased upper motor neurons. And he identified NU9 based on its ability to reduce protein aggregation, SOD1 protein aggregation. So we published this paper, and we identified actually the first compound that improves the health of diseased upper motor neurons, and that's NU9. And I don't know if I can show you this, or maybe you will have to play it. There's a short video that, can you play it, do you think? I don't have any control over here. Can you? You cannot. Okay. So this was a video that was made by the publishing group Wiley, and it showed the importance of our discovery because it was cell-based and mechanism-focused. So we didn't care if NU9 improved the health of other cells. NU9 improved the health of the upper motor neurons, and it improved the health of upper motor neurons that become diseased due to two different and distinct mechanisms. Okay? So I won't be able to show you. Here it is. But I can't start it. Anyway, that's okay. Don't worry, that's fine. Here it is moving. Yeah, there's no voice, that's okay. So we really think improving the upper motor neuron health will have great implications for HSP, PLS. We have to make those neurons happy, but there was no drug and no preclinical test using these neurons. So this was the first one. And those two model systems, one of them is the SOD1 and the other one is the TDP. And as I told you, the mechanisms are different, but the outcome is the same, that the upper motor neurons degenerate. And we can directly visualize them because they have fluorescence. Yeah, so we focused our attention to ER and mitochondria, because if we improve the health of ER and mitochondria, that is going to really help the cell. And we also looked at the apical dendrites. So NU9 was actually able to improve both the ER and the mitochondria and the apical dendrite. So that was actually very good. And here it's cell-based and mechanism-focused drug discovery. So we really have to ask the neurons what they like if we really want to make them happy, right? All right, good. So maybe I should skip. You can read the paper, it's published. Basically, we give with gavage for 60 days, different dose, 20 milligram per kilogram, 100 milligram per kilogram. We look at the improvement in mitochondria. You can see how bad it is, and with the giving of NU9, it's almost to the wild-type levels. So it improves the integrity of mitochondria. It improves the corticospinal motor neuron numbers in the motor cortex. It improves the apical dendrites, so they are not like normally apical dendrites with vacuoles are like this, and with NU9 giving 100 milligrams, so you can begin to see these apical dendrites without vacuoles. So that's actually very good. You can improve the integrity, and you look at the TDP, underlying cause, you basically see the same result. And that's very important because SOD and TDP are are distinct pathologies, so they represent different forms. All right, so this is additional information to show if it also improves axon outgrowth and axonal dynamics. And does it do it better than edaravone and riluzole, which are ALS approved drugs by FDA? And yes, indeed, it's actually, maybe I can do it better. Yes, it does. So giving a Nu9 improves the axon length. And if you give it with other, like edaravone and riluzole, it's actually better than them. And most importantly, if you give it in combination with riluzole, edaravone, there's also an added effect, which is remarkable because, again, its mechanism speaks because edaravone is free radical scavenger and riluzole is acting on astrogliosis and macrogliosis. And because a Nu9 acts on mitochondria and ER, they may complement each other, and this is exactly what's happening, I think. All right. So we now form the compound. Dr. Silverman actually initiated this, and I'm very happy that we are moving forward. And I'm also very honored to have Dr. Mitsumoto in our board of directors and scientific advisory in this scientific advisory board. So our goal is to move into clinical trials, and we would like to include upper motor neuron disease patients to these clinical trials as well. So then we identified the first compound that improves the health of diseased upper motor neurons, and we think this is an achievement, an amazing achievement for drug discovery. We are developing high-throughput drug discovery platforms, and we develop a cell-based and mechanism-focused drug discovery platforms which focus on the upper motor neuron health as a readout. All right. Remember we said biomarkers are needed. So biomarkers are needed for multiple reasons. One of them is to identify which patient have the upper motor neuron disease and by which mechanism, and also the pharmacokinetic biomarkers to see the timing and extent of upper motor neuron loss. So you remember I told you that we FACS purify them, we do the proteomics, and now when we FACS purify them at different time points, we begin to realize some genes that are upregulated, for example, it's only E15, but not really, or some other genes at P30, P60, P90, different time points, okay? And when we do the proteomics to these purified neurons, we also begin to understand what are the canonical pathways that are primarily involved in different disease models. Then we also look at the metabolites, right? The metabolites that change, and these are metabolites identified from serum and plasma samples isolated from patients. And then we do something strange. We put the data obtained from mouse and data obtained from human, and we said, are there any common ones? Because if there are any proteins that are really important, they are usually conserved among species. They have high homology, right? And guess what? Some of those proteins are, show high homology, and they are also secreted proteins. Ding, ding. So they may actually be either a biomarker or a pharmacokinetic marker, right? And because we know exactly which mouse model has them or which mechanism they have, it may also tell us if the patient has the same mechanism, right? So we are at that stage, and we, again, some of the longitudinal studies we have done based on the proteins that we have, and yes, we identified about 6 proteins that show an increasing profile as the disease progresses in ALS patients with prominent upper motor neuron loss. But now it would be nice to work with HSP and PLS blood samples to see if we can mimic the same thing, because if we do, that would be great. When we come to phase 2, phase 3 clinical trials for NU9, we may begin to include some of the upper motor neuron disease patients in our cohort. All right, so the biomarkers will help us understand disease mechanism, identify biomarkers that inform about the timing and extent of upper motor neuron loss. So we are working on this, and my collaborators at MGH, Paganoni and James Berry, we received a DoD grant recently, so we are actively working on this. And I think that we will be able to identify detection markers and pharmacokinetic markers. Hopefully next time I give you a talk, I'll tell more about that. All right, so then, we make progress on this, and how much time do I have? Shall I move very briefly? Maybe I'll tell the third part. And disease is rare, right? And there may not be drugs for all patients because it may be so rare that they may not respond to the drug treatment. Maybe we have to develop some personalized treatments for them. And I think gene therapy is a very valid option because we know the gene, we know the mutation. Why not give them the gene that is not mutated, right? So we have been developing gene therapy approaches to upper motor neurons for the past 12 years now. And first we started with retrograde transduction, meaning putting the virus into the corticospinal tract and retrograde transduction to the upper motor neurons. That's how we started at first. And we were able to retrogradely label upper motor neurons by corticospinal tract injections. We tried different serotypes like 2, 5, 8, 9, and so forth. And then, yes, you can literally label them. But this surgery is very hard in the human because the corticospinal tract is not in the dorsal funiculus in the human. So it's in the ventral horn of the spinal cord, and getting to the ventral horn of the spinal cord with a surgery is not pleasant. So then the better experiment would be, I'm going to fold this, is to the direct motor cortex injection. So that is much more easier, of course, than spinal cord surgery. And it is basically an injection to the motor cortex, layer 5. And I am told that these surgeries do not even require oh my God, staying in the hospital. Yes. So we tried different serotypes, like 2, 5, 6, 8, to see which one would be more selective to the upper motor neurons. And we found that AAV2 is more promising. Okay, so you can see this in the striatum. These are subcerebral projection neurons, all right? And then we looked, okay, who are they? Are they really who we want? And can we improve transduction efficiency by including some mutations to the capsid protein? Capsid protein mutations are important because they improve the timing of docking and selectivity and so forth. We tried different mutations. We tried different promoters. I've been like, we tried everything almost. It's like it took us 5 years to publish that paper, and so we found that using like CBA promoter, we can get 60-70% transduction efficiency. Okay, if you think about this, 60-70% is really high. You know why? Because if there was no selectivity, the percent distribution of upper motor neurons in the motor cortex is less than 1%. Okay? So now, out of all the cells and neurons transduced, 70% are upper motor neurons. Pretty good. Because if there was no selectivity, it would be less than 1%. So we improved it about 70 times. So now we actually, we published another article in Nature Gene Therapy. Now we find that upper motor neurons are indeed targets for gene therapy. Their degeneration is not dependent on spinal motor neural loss, and we have a gene target, UCHL1. And we made the cover. There was review written about this, and I think we are getting there to develop gene therapy to upper motor neurons. And so far, we have two gene targets, and also it could be that each patient has their own target, their mutated gene. So basically, we find that they have, apical dendrites all vacuolated. And when we treat them with our treatment, with our genetic treatment, we improve their apical dendrite. And this is a pathology that we see in ALS cells as well. So, the treatment improves the soma diameter, the apical dendrites. So this is actually pretty good. It almost like in the wild type, you don't have any vacuoles. In the SOD, you have a lot. And now with your gene therapy, you reduce this to this. That's very remarkable. And we do this in the TDP as well. So even though this is a different disease model, so it works in both of them. And we look at the protein aggregation and so forth. But I want to emphasize this. So I received Spastic Paraplegia Foundation grant to be able to do some of this research. And without collaboration, you can't do anything, right? And I actually collaborated with Dr. Nicholas Hatsopoulos at University of Chicago. So he's working with monkeys, macaque monkeys. And we have done the surgeries, not in the mouse, but this time in the macaque monkeys. And that was remarkable, and I'm very thankful for the support that we received from the Spastic Paraplegia Foundation because we wanted to know how selective is our transduction. And guess what? Okay, look. This is the macaque monkey cortex, okay? This is injection, and do you see these large black dots here? These are the upper motor neurons here. And this is layer 5. And you can see that not all cells in the cortex are transduced. It's mainly this layer 5 large upper, look at this, beautiful. And look at here, for example. And this is the contralateral side, which means the other side of the cortex, which didn't have the virus, right? It's clean. And here, it's basically these large neurons. And this is the injection site. This is where the needle was. Like, this is the highest concentration of the virus. And if there was any non-specificity, like, you would actually have many, many brown cells here. But even that, you actually have these large bed cells. I think this is remarkable. So we actually have our patent application now. So our goal is to go, design the viruses based on the patient. And we have done astrogliosis, microgliosis on the tissues, so there's really no reaction, prominent reaction. So we think it will be possible to develop clinical trials with patients. Let's say you have this mutation, okay, maybe we can give the correct mutation to the patient with one-time injection to the motor cortex and give the upper motor neurons the gene that they want. And then hopefully by time the neuron will say, oh, that's what I needed, thank you so much, because that's the mutated gene that causes the disease. If you give them the healthy gene and the healthy protein, maybe all the players say, hey, Jody, welcome back, we were waiting for you, and then whoop, they start playing their games, right? So that's the goal. And now we identify novel targets. We have 2 targets for now, and then of course each patient has their own target. And I think we will be able to develop selective transduction to upper motor neurons. It still works in the works. And but grants like Spastic Paraplegia Foundation grants are very vital for work. And if I say, when I started, I say, why there's still no treatment? Well, I think there soon will be. And we didn't know. Well, I think we know a lot more now. And then there was no effective drug discovery for, well, we discovered NU9, the first compound that improves the health of the upper motor neurons. We are developing platforms for more drugs to emerge, and we are working with other companies as well because they have their compound of interest, their favorite compound, and we help them determine whether their favorite compound also makes the upper motor neurons happy. And we are identifying biomarkers, both diagnostic and pharmacokinetic. And for gene therapy, I think we are developing ways for selective transduction. And I emphasize selective transduction because you don't want to mess up others while you're fixing the upper motor neuron. Because once you give the gene, it's there, all right? And then the patient is going to have it for 10 years, 20 years, 30 years, 40 years. You don't want them to develop other diseases down the line. So you want very selective transduction. And we identify novel gene targets. And that's why we wanted to initiate this upper motor neuron program at Northwestern, because our focus is to build a consortium with the goal of developing therapeutic strategies for upper motor neuron diseases, for translational medicine, for personalized medicine, and for long-term and effective strategies, because we think the time is now, and science leads the way to the effective solutions of the future, and we have to lay the pathway for that to happen. And again, I'm very thankful to Spastic Paraplegia Foundation, many other foundations, and we received grants from NIA, NIH, and I'm the scientific director of Along Swim. And of course, we have a very active group in the lab, but we always welcome hard, smart, dedicated people who are interested in upper motor neurons. Please send them my way. And I thank you very much, and I'm happy to take questions. Please note my email address, and on Twitter, on Facebook, and my website, lab website. Please visit them, and let's follow each other on, Facebook, Twitter, social, media, and let's stay friends. Thank you so much.
Hey guys, if we're going to we got time for a couple of questions, but I also want to remind you that these presentations are being videotaped with the slides, and they will be available soon on the SPF YouTube channel. A little bit later, you guys know where that is. You go to the website, you click on all those little things, you find your way through there, or go to YouTube, type in Spastic Paraplegia Foundation, you'll find all the things we've done, other presentations in the past, and then the new ones from this conference. So somebody have a question real quick.
Oh, may I say
yes, ma'am, please.
I think I have overused my time. It's okay not to have questions, to stay.
You okay?
Do we have time?
We got a few minutes.
We do? Okay.
We'll do a couple minutes, real quick here, one or two. I'm going to start here, and I'm going to work towards Ray. Here we go.
Will you be enrolling patients in clinical trials? And how do we get the information for
Yes,
that?
for NU9, phase 1 clinical trial is not going to have any patients. It's going to be healthy volunteers. And our goal is to start that next year. And for phase 2, I am on a time crunch. I need to get those biomarkers working, okay? So that's why I'm trying to have collaborations with many colleagues, and I want to make sure that the biomarkers that I have identified with a handful of samples also holds true, and so that we can have a cohort for upper motor neuron disease patients. And so that I don't know, and it will depend on the advice of the advisory board, but the goal is to include them maybe in phase 2, maybe in phase 3, but that's our goal.
Another question. Do I see a hand? One over here. Fantastic. Hi, Mary. Excuse me.
Did I understand correctly that it has been already approved, FDA approved for ALS?
What's it?
Has the NU9 already been approved
No.
for ALS?
No.
Okay, I misheard that then.
No, it has not been. So these are preclinical studies. We are trying to initiate phase 1 study next year. So it has, I hope God hears your words. I hope it's going to be approved, but has not been approved yet. We are trying to move into the clinical trials.
And actually, the first question makes me think of another. Explain to me how you're going to use healthy patients for the test, not HSP patients. Why healthy?
Okay, so phase 1 study is a toxicology study. It's an extension of the toxicology study that we have in different mammals. So we have to make sure that this compound, first of all, is not toxic at all. Okay, so that's why rather than using patients, they use healthy volunteers. They ask them, could you please volunteer for this? And they collect blood, urine, they look at liver measures, they look at blood counts. So they try to see if there is any adverse effect, if there is any toxicity. So once you pass the toxicity study, which means this compound is not toxic, then you move to phase 2, which means now you can enroll some patients, and it's an efficacy study. So then now you want to see what is the best dose, what is the route of administration, how long can we give, how much can we give, and do they respond, is there any outcome measure, can we see if it is improving? So it's, again, an experimental trial, but it is just to get more information on how to best treat, how to best modulate, how to best approach. Then phase 3 is the major one. Then phase 3 says, okay, we now know it's not toxic. We know now that it's efficacious at this modality, at this concentration, so we know. Now we say, all right, everybody jump in. So then everybody comes in and say, all right, okay, is it working or not? So then for the phase 3, the statistics is very important. But for phase 3 to be successful, the inclusion criteria is very important. The homogeneity of the population is very important. And your outcome measure is very important. What is it that you're going to measure that's going to tell you that this is successful, right? So that's why we need to have the pharmacokinetic biomarkers, which would tell us the timing and extent of upper motor neuron loss, so that we could tell, all right, it's going down, it's going up, it is responding, it's not responding. But of course, there's clinical observation, ALS-FRS scores, or what Dr. Morimoto has developed, the ALS-FRS scores for the upper motor neuron disease patients, right? So there's clinical observation, clinical quantitation, but of course, some outcome measures as well.
So is it phase 3 that you would need HSP and PLS people?
I don't understand.
Is it phase 3 that you would need HSP and PLS people?
So it's hard for me to say right now, but I would imagine. But of course, no definitive answer. I don't know. Yeah.
We're going to do one more because I'm standing right next to Mary.
Well, thank you, Dr. Ozdinler. You have an amazing, I always feel optimistic after you speak. and you have a way of taking very complex subjects, and even dinosaurs like me can understand it, sort of, kind of. So I'm going to reveal my ignorance, though, when I ask this question. And that is, within SPF, we often talk about gene mutations. And I know your research is about upper motor neurons. When we talk about different genetic therapies, and it's targeted to a particular gene mutation, would that mutation still exist in other cells, not upper motor neurons?
Excellent question. All right. All right. This is a very smart question. So now, look, when mutations occur, right? Every cell or every neuron has the mutation, right? If it's a somatic mutation, but then the upper motor neurons degenerate. So then they ask, if astrocytes, microglia, blah blah blah, if they all have the mutation, how come the upper motor neuron dies? So this is the question of the selective vulnerability. Okay. So I have an answer to that. I don't know if you like it or not. But imagine pebbles. I don't know who played pebbles when you were a kid. You know, we had little round glass balls, pebbles, different colors, right? Marbles. I'm sorry, marbles, I forgot, yeah. Marbles, yes. So we had different marbles, right? Imagine that in this room, marble is our favorite game. But we all like different color marbles, okay? So let's say my favorite is pink, yours is blue, yours is purple, yours is black, yours is white. Like, we all have our number one choice is a different color. Our number two choice is a different color. Our number three choice is a different color. But we all like playing marbles, okay? Imagine that. So which means you have the same genes, or we have the same, like marbles are the genes, we have the genes, different, genes, we have them in our pocket, but we express them differently. Like a GABAergic neuron expresses GABA, and it's an inhibitory marker, but an excitatory neuron, it has the gene, right? But it doesn't express because it's not a GABAergic. So just because you have the gene, just because you have the mutation, does not mean you are expressing that gene, you are making that mutation, okay? So coming back to the marbles. So we all have different marbles, and the production facility says, I'm not making red marble anymore. Done. Red marble, no more. Okay. So now, we are all in the room. Who's going to cry? The person who thought that red marble was their primary marble, that without that marble, you can't really play any game, that your favorite marble is red, and they're not making it anymore. You're out of the game. But I don't have red marble in my pocket. I don't care. And I continue to play. Do you see what I mean? So it's not really, oh, they all have the mutations, how come they're okay? They don't express it. They don't express the gene. They don't have it. They don't care about the gene. So the upper motor neurons, they care about that gene. They care about that protein so much. They overproduce. They overproduce a lot. But when it is mutated, it says you're not doing what you're supposed to do, and then it's making more, and you're not making any more, and then it aggregates, and it's like you have all these proteins that's not making what it is supposed to make, and you are not doing the job. Like, what am I going to do now? So it's added stress. But other cell just next door, what's your problem? What's your problem? I don't see a problem. Yeah, because they're not expressing that gene. They don't need it. So that's the thing. I think this mutation, human patient mutations, allowed us to reveal that, because what you express eventually makes who you are at a cellular level, right? So even though we all have access to the same genome, right, or all cells have access to the same genome, they don't express the same genes to the same level. So then we try to see who likes which gene and what games do they play with that gene. And if that game is all falling apart, can we give them the toys to play the game again? So that's the answer.
Well, thank you very much. We're gonna finish now and get ready for our next presentation. Dr. Ozdinler.